Hi friends, so now I'm back with my next video, and in this video we are going to talk about RNA splicing. So this is a type of post modification of the RNA which is transcribed from the DNA. So before going for the translation, in especially in eukaryotes, before going to the translation, the RNA needs to be functional, and there are some modifications that are necessary to be translated for to the protein, and one of this modification is splicing, which we are going to talk about today. So please make sure to like, share and subscribe my channel. So to watch the video related to transcription and the DNA replication, please watch my previous video in which I have talked about the transcription in prokaryotes and eukaryotes. And today we are going to talk about splicing. So what is splicing? It is a type of the modification of the nascent premature RNA, pre-messenger RNA, in which introns are removed and exons are joined. So I will talk about what are introns and exons. So here you can see a picture in which you can see three different type of exons and two type of introns. So in splicing, the introns, which are particularly the non-coding RNA, which means that they don't code for protein, and exons are something which codes for protein. So to form the mature RNA, the introns needs to be removed. So this splicing removes these introns and join the exons. Here you can see this example with the help of the spliceosome. So the intron is completely removed and different exons are joined. So this process is called splicing, and it usually take place within the nucleus. So after transcription, as you know, the transcription occur in the nucleus and translation happens in the cytoplasm. So before moving to the cytoplasm for the translation, splicing happens in the nucleus. So it happens after or concurrently with the transcription, and it is, as I have told you, it is specifically for eukaryotes before it can produce the protein through translation. So, what are introns? As I have told you, that they are the nucleotide sequences in DNA and RNA which does not code for protein. So they are removed during the this processing. They are removed because they don't code for protein. So here these introns, as you can see, in between the exons, they don't code for protein, so they need to be removed to form the functional mature RNA which codes for protein, and it can range from different size from 10 to 1000 and can be found in variety of genes and also in viruses as well. And then what are exons? Exons are the protein coding sequence that codes for protein, essential for protein synthesis. And as you know, they are the unit of protein synthesis that can code for various protein. And exons also include some of the portion of the 5' and 3' untranslated region, which contains the start and stop codons in addition to the protein coding sequence. So here is an example. So here you can see a region of DNA which contains exons, introns, different exons, different introns. When the transcription happens, everything get transcribed, all the exons, all the introns, everything. And when the processing occurs to remove the introns, the splicing, so you see these introns are removed. But apart from that, this region, which is not including like the untranslated regions, they are also the part of the mature RNA. So exons also include these untranslated regions where the introns are removed, and some of the start sites and stop sites are also included in this. So this is the example how different introns are removed and different exons are joined. So I will talk about the mechanism. How does this happens? What are the different proteins involved in this? So what is RNA splicing? Basically, it is carried out by a large complex which is called spliceosome. So what is the spliceosomes? It is a combination of different protein, and then the RNA get mixed with them, like function with them to form the RNA protein complex, which are called snRNPs, small nuclear ribonuclear protein. And there are different type of the snRNPs, And in splicing, there are 5 different type of snRNPs which are involved, which I will explain you their function just to give you a brief idea. U1, U2, U4, U5 and U6, and what is their function I will explain, but just to let you know. So U1 detect the 5 prime splice site. So what does that mean? So here you can see there are 2 exons, right? And then there is in between the intron. So at the start of the intron, there is the splice site which is GU, which is called the 5 prime splice site. And at the 3 prime, the end of the intron, there is this AG which is called the 3 prime splice site. So you, and in between of these there is the branch point which is closer to the 3 prime splice site, which contains A, adenine. So different snRNPs, how they function. So the first snRNP U1, it binds to the 5 prime splice site, and then U2 bind to the A prime splice site. So as you can see, the U1 bind to the 5 prime splice site, and U2 it binds to the branch point, and then it helps in moving this branch point towards the 5 prime splice site. So this complex, the binding of U1 and U2, is called the A complex. And now we move to the another complex, which is U4, U6, U5 binds to this complex and forms it the B complex. So all the 5 snRNPs bind to the introns. And now what is the function? So U6, it is an inhibitor of the U4, it's an inhibitor of the U6. So in the next process, the U1, which is bind to the 5 prime splice site, frees the 5 prime splice site and takes the U4 with it. So now this 5 prime splice site is free, which can be cleaved, and it binds to the U5 complex. So here you can see the splice site, it get cleaved and it binds to here, right? And then it brings closer the exon towards the U6 slide site. And next there is another cleavage of the 3 prime splice site, and then this lariat complex of the intron and U2, U5, U6 is released, and the exons are joined by the trans-esterification reaction, in which the carboxyl group and the OH group undergo the reaction and forms the bonding between them, and then 2 exons are joined. So this is in general how the splicing happens. So there are different kind of introns, as we have talked about the major complex, which include the GU, AG and the branch point A. There are other introns which does not require this complex, so they are called self-splicing introns. So what does they do is, instead of requiring the spliceosome complex, they have the some specific sites, and at this specific site G is present, and it functions exactly the same like the branch point in spliceosome A, and then it reacts with the 5 prime intron, right, at the starting of the intron, and then this get cleaved, and then this is called the transient intermediate, and then this OH group reacts to the attack this the start of the next exon, right, or the end of the intron, and then this is released as linear. And then there are other type of introns where instead of G there is A, exactly the same as spliceosome. A at the mid at the specific site, which functions like same. It attacks the start of the intron, and then forms a something like lariat and released, and 2 exons are joined. So as you can see, in this kind of introns, there is no need for the spliceosome, but adding the spliceosome increases the rate of the reaction because the enzymes are involved to perform this reaction. So the reaction is fast as compared to the self-splicing introns. So until now we have talked about splicing where the introns are removed and exons are joined. So there is something called alternative splicing. So as you know, suppose this is a DNA strand which contains 3 exons, and when it get transcribed there is different splicing happens. So in general, what splicing means is removal of introns. So what we expect is the joining of exon 1, exon 2 and exon 3, which is the usual case. But what happens in the alternative splicing, that sometimes some of the exons they are skipped during the splicing. So instead of joining all the exon like exon 1, exon 2, exon 3, what happens is exon 2 also get removed while removing the intron 1 and intron 2. And then what we get is exon 1 and exon 3 are joined. So as you see, they come from the same pre-mRNA, but the product is different. So in one of the product you have 3 exons, in the another one you have 2 exons. So the protein will also be different. So in this way, one mRNA it can code for like one gene, it can code for different protein based on the splicing and other modifications as well. So I hope that splicing is clear, and if you have any doubts please let me know in the comment section, and please make sure to like, share and subscribe my channel. Thank you.